Welcome, everyone. My name is David. I'm going to be doing the presentation of the lecture on Intro to Biochemistry. So I'm going to be going over bonding interactions, amino acids, and enzymes. So like I said before, we are recording this lecture to be posted on YouTube. So by being here, you are consenting to us recording the lecture, and in case your name or your picture is in the lecture, if you want to change it to maybe just your first name or change your profile picture, you can do that right now. Okay. And if you guys want to read this, this will be in the lecture if you wanted to go look at it after. So just a quick description on the course. We are doing a 15-week MCAT course right now. We're presenting lectures on specific topics on Zoom every Thursday, Friday, Saturday at 8:30 p.m. EST. And then every Sunday on Zoom, we're also doing office hours. If you have any specific questions or problems you want to go over, you can tune in at 8:30 and we can help you go over them. And here are just some links if you want to check them out. So we do have a group meet chat if you want to join and you can ask questions. The tutors or the other students can help you out. We also have a YouTube video where the live recording will be posted, and then we also cut up the videos into little like 10-minute clips if you want to go over specific topics. And we also have a website, socraticmed.net, where we post a lot of information and also the YouTube clips as well. And then we also have an Instagram, and if you want to follow that, they give you daily updates. Okay. So just some learning objectives that I'm going to go over today. I'm going to go over all the 20 different amino acids for proteins and the structures. And then I'm going to also go over the interactions between the different amino acid residues in the polypeptide chain. And finally, I'm going to go over the different types of enzymes and the specific functions of each enzyme. So first off, I'm going to go over the amino acids. So amino acids, they're the basic building blocks of proteins. Each one combined in a chain will produce a specific protein. When you combine a long chain of them through peptide bonds, they form a polypeptide. And the different groups on a amino acid are first off the amino group, which is NH2. So that's this blue group right here on the left side. And then also the carboxyl group, the COOH group, which is on the right side over here in red. And each amino acid will also have a hydrogen attached to it. So that's this black H at the top. And then finally, the side chain, or otherwise known as the R group, that's the group that's different in the amino acids. So depending on which R group you have, this will create a different amino acid. And you can recognize the different amino acids by the different side chains attached to it. And then this carbon in the middle that's bonded to these four groups, it is known as the alpha carbon. So in this lecture, I'm going to go over the proteinogenic amino acids. So these are the 20 amino acids in our body that are encoded by DNA and then RNA, and then finally made into proteins. They're called the proteinogenic amino acids because these are the 20 amino acids that our body uses to make proteins. There are other amino acids in the world, but they won't be covered on the MCAT. So all 20 of amino acids, out of the 20 amino acids, 19 of them are optically active except for glycine. And that's because glycine has a hydrogen as the R group. So because it has two hydrogens attached to it, it's not optically active. And then the amino acids that we're going to go over today, the 20 of them, they're all L amino acids. So in an L amino acid, the amino group, so this NH3 plus, is on the left side in a Fisher projection. As you can see on the right, the amino group is on the left side, and that creates a S absolute configuration in all 19 of the amino acids, in 19 of the amino acids, but the cysteine is the only one out of the 20 that has an R configuration because the R group is a CH2SH, so the sulfur actually has a higher priority and gives it a R configuration instead. So now I'm going to go over the 20 different amino acids. So they're split into different groups. The first group I'm going to go over is the nonpolar and non-aromatic amino acids. So nonpolar, they're usually made of the carbon and hydrogens because they're aliphatic chains, and they're also non-aromatic because they're not conjugated. So the first amino acid is the most basic one, glycine. Each amino acid has a 3-letter code and then also a 1-letter abbreviation. You're going to have to know these for the MCAT. So for glycine, the 3-letter code is GLY, and then the 1-letter code is G. So on the right, as you can see, each amino acid has the same structure that's in black, but then this green circle right here, that is the R group. So that's the group that will change between the different amino acids. So for glycine, it's the most basic one. Yes, it has a hydrogen. And then for alanine, the 3-letter abbreviation is ALA, and then A is the 1-letter abbreviation. And this has a methyl group as the R group. So as I'm going through these amino acids, the nonpolar chains, they're going to get a little bit bigger. So valine has 2 more carbons, valine VAL, and then V as the abbreviation, has 3 carbons. You're going to have to know the basic structures of the amino acids and the abbreviations as well for the MCAT. And then leucine is the next one, LEU, and L as the 1-letter abbreviation. It has 1 more carbon attached to it. And these are all nonpolar, non-aromatic ones. And then finally, isoleucine, ILE, and then I as the isoleucine. So don't confuse this capital I for an L. All the 1-letter abbreviations, they're capitalized. So that this is an I, not a lowercase l. That has 1 more carbon. And then methionine, MET, this is this is the first amino acid incorporated into a protein. The 1-letter abbreviation is M, and this is a structure for it. It has a sulfur in the carbon chain. And then the last nonpolar, non-aromatic amino acid is proline, PRO, P, and you can always identify proline because of this 5-carbon ring in the structure with the amino group. So it forms a intramolecular ring, and because of the ring, proline, it's usually at the end of a chain or in the turns and loops of the alpha helix. So those are all the nonpolar and non-aromatic amino acids. Now I'm going to go over the nonpolar and aromatic amino acids. So these amino acids, they have a conjugated system in the R group. So the first one is tryptophan, TRP, and this is the first weird one. The 1-letter symbol is W. And you can identify it by these 2 conjugated rings. And then phenylalanine, PHE, and this is also weird too. It's F. You can remember it because pH makes an F sound. And this has a phenyl group attached to it. And lastly, this is a polar and aromatic amino acid. So it's polar because of this hydroxyl group at the end, this OH. It causes the amino acid to be polar because it's not just carbons and hydrogens in the chain. This OH hydroxyl group adds polarity to it. So tyrosine, TYR. It's also weird. The one letter symbol is a Y. And then, yeah, so it's polar due to the OH on the benzyl group. And then these amino acids, they're polar but non-aromatic, so they don't have the conjugated ring system. So the first one is serine, SER-S, and it just has the carbon attached to the hydroxyl groups. And the next one, threonine, THR-T. This has just an extra carbon attached to the R group compared to serine. And then these are also polar but non-aromatic, but instead of hydroxyl groups, they're amide groups. So first one is asparagine, ASN. This is also weird too. N is the single letter abbreviation, and it's an amide group as the side chain. And then glutamine, it's similar except it has one more carbon in the chain. Glutamine, GLN, and it's also weird. It's Q. So I usually remember glycine as G, and then this Q, it kind of looks like a G. So try to remember glutamine as the one that has Q because the Q kind of looks like a G. So later on, I'm going to go over how the amino acids either lose or gain H pluses due to pH changes. But I'm just going to go over it really quick over here. So these amide nitrogens in the side chains, they don't lose or gain protons due to pH changes, but the amino group in the actual amino acid, these NH3 pluses at the top, they do pronate or depronate depending on the pH change. And finally, the last polar but not non-aromatic amino acid is cysteine. Cysteine, CYS. C, it's different than the other amino acids because it has this thiol, and thiol or SH, the bond is weaker than a bond formed by a hydroxy group, since sulfur is a larger molecule than oxygen, so it's less electronegative. So the bonds it forms due to the SH is weaker than if you were to form like a peptide bond with an OH to amino group. But cysteine is interesting because it can form disulfide bridges. So when you have two cysteines close to each other, it can form cysteine, which is the result of the disulfide bridge. So at the bottom, you can see two cysteines combined together and the two sulfurs bond to each other. But you can break this up using a denaturant that oxidizes this bond and reduces it, oxidizes it in order to break apart the disulfide bridge. So cysteines, the disulfide bridges, they contribute to the tertiary structure of proteins. And then, so acidic amino acids, these are one group of amino acids that that the R group can pronate or depronate based on pH changes. So when the side chain is deprotonated, it can have a negative charge. So the first one is aspartic acid, ASP, and the one letter abbreviation is also weird. They're running out of letters, so they use D. And so this on the right is the deprotonated form. As you can see, the O minus in the green circle. So that carboxylic acid was deprotonated. So the O has a negative charge. And when it's deprotonated, it's called aspartate. So on the MCAT, they'll either use aspartic acid for the protonated form or aspartate for the deprotonated form. And I'm going to go over this more later, but just want to let you know that the pKa of this side chain of the aspartic acid is 3.90. And then glutamic acid looks similar to it. It just has an extra carbon in the chain. So the abbreviation is GLU or E. So just remember that D and E, the two letters close to each other, they're both acidic acids, acidic amino acids. And similarly, it's called glutamate when it's deprotonated and glutamic acid when it's protonated. So on the right is the deprotonated form with the O minus. And the pK is similar. It's a little bit higher than aspartic acid. It's 4.07 for the R group. And finally, the last group of amino acids, the basic amino acids. So these are the amino acids with side chains that when protonated, it can have a positive charge. So the first one is lysine, LYS, and the letters K. I try to remember as K is the letter after L, lysine. So leucine is L, but lysine is K. And the pKa of the side chain is 10.54. And then arginine, it looks different than lysine. So it has these 3 nitrogens in the side chain. So the 3 nitrogens, it's able to delocalize that positive charge once the side chain is protonated. The positive charge can be delocalized between those 3 nitrogens. So if you move some bonds around, you can move the positive charge to any of the 3 nitrogens in that side chain. And the pK for that side chain is 12.48, so it's pretty high. And arginine is ARG or also R. So arginine kind of sounds like R, the letter R. That's how I kind of remember that. Okay. And then the last amino acid is histidine, HIS, H. So it has this ring called the imidazole ring. The positive charge when protonated is delocalized between those 2 nitrogens in the ring. And the pK for that side chain is 6.04. Any questions before I go on to a problem? Where is phenylalanine? So phenylalanine is what's at the front. Oh, past it. Yeah, so it's phenylalanine is nonpolar but aromatic. So it's nonpolar because of all those side chain, those carbons in the side chain. It's hydrophobic, and then but it's aromatic because of that conjugated benzene ring attached to it. The conjugated phenyl group. Which nitrogen is deprotonated first when in an environment? I'm not really sure what you mean by that, Opal.
I think you said.
Sorry, Opal, you were echoing a bit.
I think you said it's the nitrogen on the R group and not the one on the amide that gets deprotonated? Did you go over
Arginine?
that one? In general, you said there's one of the nitrogens get deprotonated first and not the other one.
Yeah, so usually it's one of the two at the end just because there's less steric hindrance, but it
Okay.
can be resonated between the three nitrogens, the plus charge.
And is that for all amino acids or just these particular ones?
Uh, well, this is the only, well, arginine, lysine, and histidine are the only ones that can become, have a positive charge when protonated.
Oh, okay. So those first.
So
All right, good.
the other
Mhm.
amino acids, they can, the amino group and the carboxyl group can be protonated or deprotonated, but the side chains can't be.
Can or cannot be.
So any amino acid besides the basic or acidic ones, the side chains can't be protonated or deprotonated, but the amino and the carboxyl group of every amino acid can be.
Okay, thanks.
No problem. Yeah, so for this, I recommend, so what I did was on my phone, I saved a picture of the amino acids as my background. And then every time I opened my phone, I just saw that picture of all the amino acids. And that kind of like embedded it into my brain. Is that, if anyone wants to try that. Any other questions? I think I kind of gave everyone saw the answer, but any questions before I go on to this problem? Okay, if not, I'll start the poll. Oh, sorry, hold on one second. The poll's not working. Give me 1 second. I'm going to stop the screen share for a second while I figure out this poll. Sorry about that. Okay, sorry about that. Yeah, so if you want to take a minute to look at this question and answer it. Opal, can you not see the poll? I'm not sure. Poll does look a little different. I'm not sure why you can't see it. if you want, you can just put your answer in the chat, or just wait until I go over the answer to see if you got it right. Just keep like a mental note of your answer. All right, I'm gonna give everyone about like 15 more seconds if you want to get your answer in. Just take a guess if you don't know. All the answers are anonymous. Okay, I'm gonna end the poll now. So it looks like most people picked choice B, and so that is the correct answer. So which of the amino acids has a net charge at physiological pH? So glutamic acid is the only acidic amino acid on here. So so the side chain of the glutamic acid, it will have a negative charge because of the carboxyl group as the side chain. So it will have a negative charge once it's deprotonated. Any confusion on this question? Okay, all right, I'll move on. So I'm gonna go over if the amino acids are hydrophilic or hydrophobic. So hydrophobic amino acids, those are usually the non-polar ones. So it usually has an alkyl chain as its R group, because of the large amount of carbons and hydrogens in the side chain without any polar molecules such as like oxygen, nitrogen, sulfur. Because of that, they'll be hydrophobic and they'll try to avoid water. So that's why they're usually found in the core proteins away from the aqueous environments that the proteins are in. On the other hand, hydrophilic amino acids, these are the basic amino acids with the amino group, the acidic amino acids with the carboxyl group, and the amide amino acids with the amides as R groups. So those are able to interact with the water and form favorable interactions such as hydrogen bonding. And they're usually found on the outside of the proteins, on the exterior where they have more access to the aqueous environment. And so the rest are usually called neutral, so they don't really prefer hydrophobic or hydrophilic environments over the other. And on the right, I've just included two charts. So the hydrophobic amino acids is this top one. You can see it's a lot of carbons and hydrogens. While the bottom ones, hydrophilic amino acids, they are usually the ones that have these polar groups, such as the amides, amino groups, and the carboxyl groups. Okay. So like I said before, the amino groups, the carboxyl groups, and the side chains of the acidic and basic amino acids, they can become protonated or deprotonated depending on the pKa of the group and also the pH of the environment. This is because amino acids are what we call amphoteric species, so they can either accept or donate protons depending on the pH of the surrounding. So like I said before, the amino groups and the carboxyl groups of every amino acid can become protonated or deprotonated, but then only the R groups of the basic or acidic amino acids can become protonated or deprotonated. And whether they're protonated or deprotonated, it depends on the pK of the group and then the surrounding pH. So when I was going over the acidic basic amino acids, I went over the pKa's of each R group, but the pK of the carboxyl group of every amino acid is around 2, and then the pK of the amino group on each on each amino acid is between 9 and 10. Usually they round to around 9. So in this chart, you see here the H hydrogen on top isn't affected, but the amino group has a pKa of around 9, and the carboxyl group on the right has a pKa of around 2. And then the variable side chain, the R group of the 20 amino acids, they're either unaffected if they're not basic or acidic amino acids, but if they are, then the pKa of those groups determine if they're pronated or depronated, and also the pH of the environment. So how do you determine that if it's pronated or depronated? So if the pH of the surrounding environment is less than the pK of the functional group, then the functional group is pronated. But if the pH is greater than the pK of the functional group, then the functional group is depronated. And then there are special cases called zwitterions where the amino acid has multiple charges, but the charges neutralize each other, and it results in a neutral molecule. So you can see in this diagram on the right is at high pHs, the functional groups are depronated. So the carboxylic acid has a negative charge, while the amino group does not. It has a neutral charge, and because of that, there's a negative charge on the amino acid. And then on the left side, you can see at low pHs, both groups are protonated. So the amino group is protonated and has a positive charge, while the carboxyl group is protonated and has a neutral charge. And because of the positive charge on the amino group, the whole amino acid is positive charge as a result. So then in between the two pKs, you can have a zwitterion where the amino group is positively charged because it's protonated, while the carboxyl group is deprotonated and has a negative charge. So at certain pHs, this can happen, and because of the positive charge on the amino group and the negative charge on the carboxyl group, these two cancel out, and you have a neutral charged amino acid, even though there are two different charges on the molecule. So the positive charge and the negative charge on that same amino acid cancel each other out, and you have a neutral molecule, and that's called a zwitterion. Okay. Any questions on this before I go on to a problem? You can either unmute your mic or just put it in the chat. And also, if you have any questions, you can also put it in the chat, and Mark, the standby tutor, can help you out.
I have a question.
Yes.
So my question is more based on the topic in general. In the MCAT, is like in regard to the topic of amino acids, is it solely based on the 20 proteinogenic amino acids, or have you ever seen the MCAT test other amino acids?
So usually it's related to the 20 amino acids, but the MCAT is random. They could give you like a random amino acid that you never heard of, but then you have to relate it to the amino acids that you that are on the MCAT, so the 20 that we went over today. So you're not required to know any other amino acids, but the MCAT, they're very random. They could give you an article about like another amino acid that you've never heard of, but you're not expected to know anything about it. You just have to know everything about the 20 on this lecture and use that to relate to the passage, if they do decide to do that.
Okay, thank you.
No problem.
Hey, David, are
yes.
we expected to know which amino acids are part of the catalytic triad? You heard of that? You know how we know proline is like cysteine for form disulfide bridges and proline form kings. like they'll have like histidines. A few of them are like a part of this. Our histidine is in the active sites of most enzymes because are we expected to know which ones are?
You should be able to differentiate between them. You don't need to know like the specifically how it does it, but you should be able to know. Yeah, you don't need to know like I don't think you need to know the name catalytic triad, but you should know the function of the 3 amino acids. Those 3.
Which 3? Which forms form the triad? Histidine and which ones?
Yeah, so histidine is in a lot of active sites enzymes.
Okay, and which other ones?
Yeah, so proline, like I went over because of the ring, it forms like kinks in chains. So usually it's at the end or the beginning of chains, and also at the in the turns of the alpha helixes. And then and then cysteine, it forms the disulfide bridges, and that contributes to the tertiary structure of proteins.
I'll go look it up. I think serine was a part of the triad, but it's okay. I'll research it. That's fine.
Yeah. I don't think you need to know like specifically the triad that you mentioned, but you should know if each what each amino acid does, if it has like a specific
purpose
function. Yeah.
Okay.
Any other questions before I post the problem? Okay. All right. So try this question. I'm going to start the poll. Okay. I'm really on the poll. Just take a few minutes and guess if you're not sure. Okay, about 20 more seconds if you want to get your answer in. Okay, I'm gonna end the poll now. Okay, so everyone picked D, and yep, that's the correct answer. Lysine. It's one of the basic amino acids. So it's very important to know the type of amino acids each which group each amino acid belongs to, because on the MCAT, a lot of times they'll give you like a long chain of amino acids and tell you to figure out the net charge of this chain, and that's when the basic and acidic amino acids are really important to know. All right, any questions before I move on? All right, I'll move on. So the next thing I'm going to go over is the bonding interactions between the amino acids. So one of the most important ones is hydrogen bonds. So you can have like intramolecular hydrogen bonds between like large molecules, but for amino acids, we know it as a strong intermolecular force between the different residues. So hydrogen bonds, they're created because of the differences in electronegativities between hydrogen and a electronegative group such as oxygen, nitrogen, or fluorine. And in amino acids, we are particularly interested in the nitrogen and the oxygen, since there are no fluorines on the amino acids. So because of the electronegativities, the hydrogen has a partial positive charge on it, while the oxygen or nitrogen has a partial negative charge. That's because the electronegativity of the oxygen nitrogen, it draws more of the electron density towards the oxygen or nitrogen, which leaves a partial positive charge on the hydrogen. So because of the partial positive charge on the hydrogen, it is able to interact with the partial negative charge of a oxygen or nitrogen on another amino acid. So as you can see in the picture on the right, because of the partial positive charge on the hydrogen will interact with the oxygen or nitrogen on another molecule that has the partial negative charge. And so in that case, we have a donor that donates the hydrogen for the hydrogen bond. So it's any molecule with a oxygen, nitrogen, or fluorine that has a hydrogen bonded to it that creates the partial positive charge on the hydrogen. And then acceptors are the molecules with either an oxygen, nitrogen, or fluorine with a lone pair of electrons that has the partial negative charge, and that binds to the hydrogen from the donor. Yeah, so in amino acids and proteins, the amino group will hydrogen bond with the carboxyl groups in the peptide backbone, and that forms the secondary structure of proteins. So the primary structure is the long chain of peptides that forms the polypeptide chain, but the hydrogen bonds between the amino and carboxyl groups of each amino acid, the interactions between them, between the different residues, they form the secondary structure of proteins. So the two different types on the MCAT are the alpha helices. On the left, you can see it's that spiral shape, and then the beta pleated sheets, which are the sheets that lay on top of each other, well, side by side in this picture. And the hydrogen bonds, they provide stability to the protein. So the hydrogen bonds, they form the proper secondary shape of the protein, and it provides stability until like a denaturant comes by and breaks the hydrogen bonds. The other interactions by amino acids are the hydrophobic interactions. So like I said before, the different hydrophobic amino acids, they'll be attracted to each other because they're trying to repel the aqueous environment. So they'll try to repel the polar molecules such as water or the other hydrophilic amino acids. So as a result, the hydrophobic amino acids will be on the inside of a protein. So they'll be in the interior of the core. And this allows more hydrophilic amino acids to interact with the aqueous environment, because the hydrophilic amino acids, they'll be pushed to the outside, the exteriors of the protein, while the hydrophobic amino acids are able to become concentrated in the interior core of the protein. So the hydrophobic amino acids will interact with each other while pushing out the hydrophilic amino acids to the outside for it to interact with the aqueous environment. And this maximizes stability of the protein, because it won't be like a huge mess of random hydrophobic and hydrophilic amino acids interacting with each other. It becomes more concentrated, where the hydrophobic amino acids are on the insides of the protein, while the outsides are more hydrophilic interactions. And hydrophobic interactions, this determines the tertiary structure of proteins. So this forms the tertiary structure of proteins, which is usually the final form of the protein, unless it has a quaternary structure. And so on the other hand, there are hydrophilic interactions where the hydrophilic amino acids, they're found, like I said, on the exterior of the protein. And this is done in order for it to interact with the polar and aqueous molecules in the environment. So most proteins, they work in the cytoplasm or other aqueous environments. And as a result, the hydrophilic amino acids are on the outside, because it shields the hydrophobic residues from the aqueous environment. So like I said, this will concentrate the hydrophobic amino acids in the center, while the hydrophilic ones are on the outside, and this is done to increase stability of the protein. So since the hydrophilic amino acids are on the exterior of the protein, it creates what we call a solvation layer around the protein. So this is a layer of water molecules, and this is done to increase hydrogen bonding. So the water is forced into a specific arrangement that maximizes hydrogen bonding with the hydrophilic amino acids. And this also contributes to the tertiary structure of proteins. Any questions before I go on to a problem? You can put in the chat or just unmute. Okay, all right, I'll move on to the question. So which of the following is most likely to be true of nonpolar R groups in an aqueous solution? I'm going to give everyone a few minutes for this. Let me start the poll. Okay, all right, I'll give everyone a few minutes for this. Sorry, Opal, I think you're still unmuted. If you just unmute.
Sorry.
That's okay. All right, I'm gonna end the poll in a few more seconds if everyone wants to get the final answers in. I'm gonna end the poll now. And it looks like everyone clicked C, which is the correct answer. Yeah, so nonpolar R groups, they're the ones that have mostly carbon and hydrogen, so they're going to be hydrophobic. And because they're hydrophobic, they're going to be buried deep within proteins away from the aqueous environment. So, yep, everyone got that right. Any questions on this before I move on? Okay. All right. So lastly, I'm going to go over enzymes, how they function, and the different types, and then also enzyme kinetics. So what are enzymes? So enzymes function as biological catalysts that increase the rate of chemical reactions. They increase both the forward and the reverse reaction. So some enzymes, they can catalyze the forward reaction, but then also reverse reaction. So they turn the product that you just formed back into the reactant. And enzymes, they can catalyze both the forward and the reverse reaction. So they catalyze the reaction by lowering the activation energy barriers. So reactions require a certain amount of energy input, and enzymes function by lowering the amount of energy needed. So reactions can occur faster because there is less energy needed to overcome that activation energy barrier. And a unique feature of enzymes is it's not consumed by the reactions. So after each reaction, it will revert to its original form, and you can reuse it again. So it can be reused over and over again after each reaction. And enzymes can be affected by environmental factors. So enzymes are proteins, so they can be denatured if certain conditions are met. So for temperature can increase an enzyme activity up to a certain point. So usually as you increase temperature, the activity of the enzyme increases. But once you reach a certain point, if you exceed its optimal activity, then it can become denatured and you can actually decrease its activity. And then certain enzymes, they function at specific pHs. So like enzymes in the stomach will usually occur at lower pHs, while enzymes in the intestines will function at more elevated pHs. Each enzyme has a specific pH that is its optimal range. And then salinity, so it also can be affected by salt concentration. So each enzyme will also have a specific salt range that it can operate within. So how does the enzyme catalyze reactions? So first, the enzyme binds the substrate in the active site that is specific for each enzyme. So each enzyme will only bind to a specific substrate, and that substrate is determined by the active site. So the active site is the actual site that binds the substrate and performs the reaction. So once the substrate is bound in the active site, it forms the enzyme substrate complex. So that's the if you look at the bottom picture, it's the second form. So that's the enzyme substrate complex when the enzyme is bound to the substrate. And then once it performs the reaction, once it catalyzes the reaction, it will release the products and it is ready to catalyze another reaction. So it's not used up. It can start catalyzing another reaction right after. And like I said, it returns to its original shape and remains unchanged. So on the bottom, just a small diagram on how the the enzyme catalyzes the reaction. So next I'm going to go over the different types of enzymes. So there are a bunch of different enzymes. Each one performs a specific reaction. So oxidoreductase, this is used in redox reactions. So it's used in reactions that require oxidation and reduction, and it does it by transferring proteins from the oxidant to the reductant. They usually use NAD plus or NADP plus as cofactors that help perform the reaction. And usually if you see the name dehydrogenases or reductases in the enzyme name, it's an oxidoreductase. If you've gone over the glycolysis or the Krebs cycle, you'll have seen these oxidoreductases. There's a bunch of different dehydrogenases and reductases in that those reactions. And then so that's oxidoreductase. And then next one I'm going to go over is transferase. So exactly as it sounds, it transfers functional groups. So it moves functional groups from one molecule to another. So the first one I'm going to go over is aminotransferase. So exactly as it sounds, it transfers amino groups. So usually this is used to convert one amino acid to another, or move amino groups in other molecules. And then next is kinase. So there you'll recognize that there are a lot of different kinases, and they transfer phosphate groups from one molecule to another. So they'll either add a phosphate group to one molecule or take away a phosphate group from another. So it transfers phosphate groups. Kinases are very important. Be sure to remember what kinases do. They can transfer phosphate groups. Next one is hydrolase. So hydro, you can remember as water. So it breaks down a compound using water. So you might have heard of phosphatase. So they cleave off phosphate groups. So a lot of times they're used for like inactivation and activation of a molecule. So phosphatases, they'll cleave off a phosphate group to either activate or inactivate certain molecules. Sorry, Opal, I think you have your mic still on. Thank you. Okay, and another type of hydrolase is called peptidase. So those cleave proteins by cutting peptide bonds. And then nucleases, they cleave nucleotides in, sorry, that should say nucleic acids. So nucleases, they'll cut DNA or RNA. And then lipase, they cleave lipids. So those are the different the hydrolases. And similar to hydrolase, except it doesn't use water. So lyase also cleaves compounds into smaller molecules, but it doesn't use water. And like I said before, enzymes can catalyze the forward and the reverse reaction. So synthase, it catalyzes the opposite reaction. It combines two molecules into a single compound without using water. So lyase and synthase are opposites to each other. They catalyze the opposite reactions. And then isomerase. So if you've heard of isomers, they form different stereoisomers or constitutional isomers. So they rearrange bonds within a molecule, and changing the bonds of a molecule can change the whole structure and the whole function of a molecule. So isomerases, they can form various stereoisomers or constitutional isomers, depending on the different isomerases. Ligase, it's similar to a synthase, except it needs ATP to complete the reaction. So synthases, they usually don't require much additional energy, but ligases, they perform addition or synthesis reactions, but they need a form of energy to complete it. So usually it's in the form of ATP. You can find these in the nucleic acid synthesis and repair. So if you remember from DNA replication, the ligases, they seal off the DNA, so they synthesize the replication of DNA by sealing the double-stranded DNA. Okay. And so next thing I'm going to go over is enzyme specificity. So like I said before, each enzyme can only bind to a specific substrate that is determined by the active site. So each enzyme will have a different active site, and that active site will bind to a different substrate. So the first model of enzyme specificity is called the lock and key model. So this is an older model where the active site is the complement of the substrate. So the substrate fits perfectly into the active site. No changes needed. So as you can see in the diagram below, the orange triangle, that's representative of the substrate, and then the blue is the enzyme. So the substrate fits perfectly into this V-shaped groove, which is representative of the active site, and it fits perfectly to form this enzyme substrate complex. No tertiary or quaternary change is needed in order to complete the reaction. So this is called the lock and key model because the key, which is the substrate, fits perfectly into the lock without any change needed. And that model is pretty outdated now. The more accepted model is this model called the induced fit model. So substrate binding to the active site of the enzyme induces a conformational change. So the shape of the active site and the enzyme is changed in order for the substrate to fit into the active site. So as you can see here, the substrate still orange triangle, but the enzyme at first, it's the active site doesn't fit perfectly with the substrate. But after the active site, after the substrate goes into the active site will undergo a conformational change in order for the active site to fit with the substrate. So this middle molecule right here, we call that the transitional state. So that's the induced form after substrate binding. So usually this will only exist when the substrate is binded. That's why it's called the transition state. It's the state between the enzyme and the reactant, and the enzyme and the formed product. But once the substrate unbinds from the active site of the enzyme, the active site will return to its original shape. So it will return to this like trapezoid looking active site until another substrate is ready to bind to it. So induced fit, it's called induced fit because the active site is induced into a certain conformational change in order to fit the substrate. Any questions on anything I just went over right now? You can put it in the chat or unmute. Okay, all right, I'll move on. So enzymes, they're not allowed to just keep catalyzing reactions without any regulation. So there are a few different ways to regulate enzymes. So the first one is called competitive inhibition. So the inhibitor physically occupies the active site. So an inhibitor molecule will bind to the active site and not allow the substrate to bind to the active site in the enzyme in order to catalyze the reaction. So it's physically blocking the active site where the substrate binds. So that's called competitive information. They're physically competing for the active site. And the next type is called non-competitive inhibition. So this is when the inhibitor binds to what we call an allosteric site. So this is a site that is not the same as the active site. The substrate will not bind to the active site. The allosteric site is usually bound to certain molecules that regulate the enzyme. So this allosteric site, if it binds to an inhibitor, it will induce a conformational change in the enzyme, and this conformational change will inhibit the enzyme from binding to the substrate. So no matter how much substrate you put into the solution, the enzymes would not be able to bind to the substrate. And non-competitive inhibition, it can bind equally to the enzyme or the enzyme substrate complex that is formed after the substrate binds. So it can bind to either the enzyme without the substrate bound to the active site or with the substrate bound to the active site to inhibit the enzyme. And then the next type is called mixed inhibition. So this is similar to non-competitive inhibition because it binds the allosteric site also of either the enzyme or the enzyme substrate complex, but with different affinities. So depending on the inhibitor, it'll either bind to the enzyme or the enzyme substrate complex more. While the non-competitive inhibitor, it'll bind either to the enzyme or the enzyme substrate with no particular affinity. So if you place it by an enzyme or enzyme substrate complex, it will bind it will have the same affinity to bind to either the enzyme or the enzyme substrate complex. But mixed inhibition is when the inhibitor will bind to the enzyme or the enzyme substrate complex with different affinity. So it will favor either binding to the enzyme or the enzyme substrate complex. And lastly, uncompetitive inhibition. So this is also allosteric regulation where the inhibitor binds to the allosteric site, but it only binds to the enzyme substrate complex. And similarly, it creates a conformational change in the enzyme, so the substrate is not able to bind. So the non-competitive and mixed inhibition, they both bind to either the enzyme or enzyme substrate, while the uncompetitive inhibition, it only binds to the enzyme substrate complex. So those are the 4 different types of enzyme regulation or inhibition. Any questions on that before I go into enzyme kinematics? Kinetics, sorry, not kinematics.
Yes, quick question, Dave. Do we need to recognize like the graphs for these different binding, like on our the Lineberg-Weaver plot where they have those KMs?
Yeah, so you will be needed. You will need to know how to differentiate between the different inhibition on the either Lineweaver-Burke or the Michaelis-Menten.
Okay.
Okay. Any other questions? Okay. So enzyme kinetics. So like I mentioned before, there are different ways to measure the reactions catalyzed by enzymes. The first one is the Michaelis-Menten. So in this plot, the enzyme concentration is constant, but the substrate concentration is increased. So you will have a given concentration of enzymes that doesn't change, but the only thing changing is the substrate concentration. So there are a few parameters you need to know. The first one is Vmax. So that's the maximum velocity of the reaction with the given enzyme concentration in the experiment or the reaction with the enzyme. And Vmax can also be measured as Vmax equals E times Kcat. So E is the enzyme concentration, and KCAT is what we call the turnover rate for a single enzyme. So that's pretty much the rate of catalyzing the reaction by a single enzyme. So that makes sense, since if you take the rate of a single enzyme and multiply it by how much enzyme you have, you'll have the maximum velocity that you can maximum velocity of the enzyme with the given amount of enzyme concentration. And the next parameter is called Km. So this is the substrate concentration when the velocity of the reaction is half of Vmax. So the velocity is occurring at half its maximum velocity. For Km, you need to know that if a reaction has a high Km, that means it has a low affinity. The enzyme has a low affinity for the substrate, which makes sense, since if you need more substrate concentration in order for the enzyme to bind to the substrate and catalyze the reaction, then the enzyme doesn't really have a high affinity for the substrate. While if you have a low Km, that means you only need a small amount of substrate in order for the enzyme to be working at half its maximum velocity. So only a small amount of substrate is needed to reach half of Vmax. So this is a standard Michaelis-Menten plot. On the bottom, the x-axis is the substrate concentration. So this is increasing as time goes by. And then on the y-axis is negative 1/2 Vmax. So this is the measuring the velocity. Oh no, sorry. The y-axis is the reaction velocity, V. And on the bottom you can see Km. So this is the maximum. This is the substrate concentration required to reach half of Vmax. So you can see it's 1/2 of Vmax on the y-axis. So at half of Vmax, this is the amount of substrate concentration needed. And you can see as Km goes more to the right, more substrate concentration is needed to reach half of Vmax. So the enzyme will have lower affinity for the substrate, while if the Km is to the right of this Km on the chart, less substrate concentration is needed to reach half of Vmax. So it has a higher affinity for the substrate. Sorry, you said, Opal, you said repeat that. Which part do you mean? Affinity and concentration needed.
Yeah, you said to the right you need more, and to the left we need less of what?
So to the right of this Km, if Km is to the right, you see on the bottom it says substrate concentration. So if Km is moved more to the right, you'll need a higher substrate concentration. So that higher substrate concentration needed to reach half of Vmax, that means the enzyme will have a lower affinity for the substrate, because you need a lot more substrate in order for the enzyme to bind to the substrate and catalyze the reaction. And then similarly to the left, if Km is moved more to the left on the x-axis, it requires less substrate concentration. So less substrate is needed for the enzyme to bind to the substrate and reach half of Vmax. So that would signify a higher affinity of the enzyme for the substrate in question. Okay. And So the next one is called the Line-Weaver-Burke plot. So it graphs the same data as the Michaelis-Menten, but it's graphed differently. So as you can see, the Line-Weaver-Burke plot, it has a standard standard slope, has a constant slope, so it has a standard line as that's plotted, while the Michaelis-Menten has more of a curved graph. And yeah, so the Line-Weaver-Burke, it just tries to simplify the data that is graphed as a Michaelis-Menten plot. It uses the reciprocal of the Michaelis-Menten plot axis. So as you can see here, the Michaelis-Menten, it plots V, the velocity of the reaction, as the y-axis, and substrate concentration S as the x-axis. But on the Line-Weaver-Burke, it plots the reciprocal. So it plots 1 over the velocity of the reaction as the y-axis, while the reciprocal of the substrate concentration, so 1 over S, is the x-axis. And so as you can see on this graph, it's kind of hard to identify 1/2 Vmax and Km, because you have to guesstimate where the middle is, where the 1/2 of Vmax is, and then you have to guesstimate where the substrate concentration is. Line-Weaver-Burke plot simplifies it. So it shows that the x-axis is -1 over Km. So you can use the x-axis intercept to identify the Km, so the substrate concentration, to reach 1/2 of Vmax. And then y-axis is 1 over Vmax. So you can use the y-axis intercept to calculate the maximum velocity of the reaction. So Line-Weaver-Burke plot and Michaelis-Menten, they're pretty similar. It's just how they're plotted. The Michaelis-Menten is good for looking at the overall velocity of the reaction, but it's hard to determine the 1/2 Vmax and the 1/2 Vmax and Km. So Line-Weaver-Burke plot just simplifies it by allowing you to calculate it using the x and y axis intercepts. Yeah, any questions before I go on to a problem? Okay, all right, so I'll give you guys this problem. I'll give everyone a few minutes for it. All right, I'll give everyone a few more seconds to answer the question if you have it put in your choice in. I'm sorry, Opal, I think you're unmuted.
Oh
again.
my gosh, sorry. What the heck?
Okay. All right, I'm gonna end the poll now. Okay. All right, so everyone picked D, and yeah, that's the correct answer. So it's asked, this question's saying a student is observing enzymatic chemical reaction that normally takes place in human blood, and she wants to perform an experiment to see how certain conditions will affect the reaction while the enzyme is fully saturated with substrate. And it wants to know how to speed up the reaction. So in the question, it said the enzyme is fully saturated with substrate. So adding more substrate wouldn't do anything since the enzyme is already bound to substrate. And increasing the temperature may speed up the reaction. Usually it can speed up reaction, but sometimes increased temperature would not speed up the reaction. So it can denature the protein, the enzyme. And also, since it's in human blood, it probably the optimal temperature would probably be whatever the human biological temperature is, and that's around 37 degrees Celsius, I believe. And then pH, we don't know the optimal pH range of this enzyme, but we know it's in human blood, and human blood is not perfect 7.0, which is what like water is. That's not our pH of our blood, so that would not speed up the reaction. But increasing the enzyme concentration would definitely speed up the enzyme, the reaction, catalyze the enzyme, because there's more enzyme to catalyze reaction. Since this reaction is saturated with substrate, adding enzymes would react with the saturated substrate in order to speed up the reaction. So choice D is the correct answer. Okay, and I have another question. Which of the following is false concerning enzymes? All right, I'll give around a few minutes for this again. Start the poll. Okay, I'll try to answer this question. All right, I'm gonna stop the poll in a few seconds if you want to get in your final answers. I'm gonna stop the poll now. All right, and everyone picked choice A. Yep, that's the correct answer. So enzymes, they can increase the amount of product created over a certain period of time because it speeds up the reaction, but it won't change the amount of product created in a single reaction. So it'll decrease the activation energy needed for the reaction, and that speeds up the reaction, but it doesn't change the amount of product created. So over a period of time, it may create more product as a result of speeding up the reaction, but it won't increase the amount of product created in a single catalyzed reaction. And choice B, yeah, so enzymes, like I said, aren't destroyed and can be reused over and over again. And they can also increase the forward and reverse rate of a reaction if it can catalyze both. And it speeds up the reaction by reducing the activation energy of the reaction. So choice B, C, and D are all true, which makes choice A the right answer because it's false. Yeah, and that's all I have for everyone today. Any questions on the lecture, anything in the lecture or the problems? Thank you everyone for attending. Yeah, so I will post this lecture in the group meet chat, and I'll also post the live stream on YouTube later if you want to watch. Let me just stop the recording.